I'd like to present to you the results of our project on micromachining of cellular lamellae for in situ structural biology. The project was carried out within the frame of the VP4 Call 1 projects, which aimed at transformation of new technologies into services at the Instruct facilities. The consortium of our project comprised of electron microscopy facilities at CETEC, EBIC, and NISEN, and the major goal was to increase the throughput and reproducibility of the cellular lamellae preparation by the Cryo-FIB. I will now summarize what we have been focusing on at CETEC. Here is an overview of the Cryo-FIB-based workflow for preparation of the specimen for in situ structural studies at high resolution. As you can see, the whole workflow is still very complex and time-consuming, comprises utilization of several high-end instrumentations and transfer in between them, which increases the risk of sample damage and decreases the throughput of the whole workflow. We have tried to address individual steps in the workflow to increase the throughput and the reproducibility. In the case of sample preparation, we have delivered a protocol for the sample preparation of the 3 different prototypical samples for Cryo-FIB. These comprise adherent cell culture, suspension cell culture of small eukaryotic cells, and protein crystals. Optimize the culture maintenance and sample preparation and vitrification, general steps in the lamella production, and transfer of the specimen to TEM for cryo-electron tomography, and we have delivered a step-by-step protocol describing this process. In the case of Saccharomyces cerevisiae, for instance, the sample can either form small patches of the cells on the grid surface, or cover the whole grid squares with monolayer of cells based on the concentration of the cells that are applied to TEM grid. In addition, the yeast has to be blotted against non-absorbic material in order to retain the cells on the grid. We have designed a blotting pads which can be easily 3D printed from flex material, which is easier to access than, for instance, the PTFE pads, which are another option for non-absorbent blotting. Following the success of the vitrification, the sample is transferred into the small dual beam microscope. The instrument constitutes a scanning electron microscope column and the ion column, which focuses the current of gallium atoms on the sample. Preparation workflow consists of a localization of the cells, ideally in the center of the grid square, and gradient removal of the cellular material by scanning with FIB in order to retain thin cellular section, which is then electron transparent for TEM. First, we inspect the grid quality, check for sample distribution and surface contamination. Then the surface of the grid is spattered, coated with a protective layer of GIS. The additional metal coating metal sputtering increases the sample conductivity and cures the GIS layer. After that, we start milling the rough lamella with relatively large current and gradually decrease the lamella thickness and width together with decreasing milling current. The final polishing step then results in a 100 to 200 nanometer thick lamella. For automation, and therefore we have developed and are now testing software for unsupervised lamella milling. Ideally, we want to end up with a lamella with unperturbed GIS layer on the front edge and a uniform thickness over the lamella surface. Significant sample contamination can result in uneven lamella milling and non-uniform lamella thickness. and the removal of the GIS layer can cause lamella cracking. We have found out that addition of 300 nanometer GIS layer provides good lamella protection, while it's still not prohibitive for efficient milling. In addition, water and hydrocarbon contamination in the microscope chamber adsorbs on the lamella surface. We have measured the absorption rate for our instrument, and we've set the lamella preparation accordingly to minimize the surface contamination. We show several sections from the tomograms collected on the yeast lamella. We can observe a paracrystalline core of the exosomes, the microtubules, nuclear pores, or membrane proteins on the mitochondrial membrane, or the mitotic spindle at high resolution, unstained specimen at near atomic resolution. To summarize, we have delivered a protocol for preparation of 3 different prototypical specimens, standardized the workflow of lamella preparation, and initiated an automation of the lamella preparation process. In the future, we will primarily focus on further automation and explore the preparation of the lamellae from high-pressure frozen specimen using, for instance, plasma PFIB. At the end, I would like to acknowledge my colleagues from the lab who primarily work on this project. It was primarily Jana Moravcová who delivered the protocols for the sample preparation, Matyáš Pinkas, who worked on the yeast samples, and Michal Babek, who is developing the software for the automation. And with that, I would like to thank you for your attention.